Good day, everyone. Our topic for today is adaptive immunity, cell-mediated immune response, which is a vital aspect of our body's defense system. We will be discussing the following topics: the types of T cells, types of T cell-mediated immune reactions, and the development and functions of effector T lymphocytes and cytotoxic T lymphocytes. Okay, let's start. Before we dig deeper to cell-mediated immune responses, let us first define what is adaptive immunity. Adaptive immunity is a specific immune response developed by the body after exposure to a specific antigen. Unlike the innate immune system, which provides a broad first line of defense, adaptive immunity is highly targeted and long-lasting. This targeted response allows the immune system to remember past encounters with specific pathogens and respond more effectively upon re-exposure. In simple terms, adaptive immunity is our body's way of fighting specific germs and diseases. It learns and remembers how to attack these threats more effectively each time they appear. Let's move on to cell-mediated immunity. Cell-mediated immunity is one of the key components of adaptive immunity. It is a type of immune response that does not rely on antibodies, but instead it involves the activation and coordination of specialized white blood cells called T lymphocytes. These cells don't produce antibodies, but they directly attack infected cells and help control the immune system. So they are essential for fighting viruses, cancer cells, and intracellular bacteria. T cells directly target and eliminate infected cells harboring intracellular pathogens like viruses, some bacteria, or abnormal cells, or the cancer cells. This targeted elimination prevents further spread of the pathogen and helps control or eliminate the threat. Cell-mediated immunity is slower to develop compared to the antibody-mediated response, but it provides a more potent and long-lasting defense against intracellular pathogens. The advantage of a cell-mediated response is that it directly targets and destroys the infected cells, including those harboring viruses or cancer cells. This makes it particularly effective at dealing with infections that hide inside cells where antibodies can't reach. Cell-mediated is a T-cell-mediated response. Its activities happens in the location of antigen-presenting tissue. Those antigen-presenting tissue refers to tissues that contain cells such as dendritic cells, macrophages, and B cells, which capture, process, and present antigens to T cells. So these cells display pieces of pathogens on their surfaces using molecules called MHC, or the major histocompatibility complex, helping to activate the adaptive immune response. So the main cell types involved in cell-mediated are the T cells. Although the speed of onset is slow, it may take a few days to develop as T cells need time to recognize the antigen, proliferate, and become activated. However, once activated, cell-mediated immunity can respond rapidly and efficiently to target and eliminate infected cells. The antigen type are the intracellular pathogens, which is a microorganism such as virus or certain bacteria that resides and replicates inside host cells, evading the immune system detection and attacking mechanisms. The other one is the cancer cells, which are abnormal cells that have undergone uncontrolled growth and have the potential to invade nearby tissues or spread to other parts of the body. These can be removed by cell lysis, or the rupture or destruction of a cell's membrane leading to its death. And the programmed cell death, or the apoptosis, it is the controlled and orderly process of cell death that occurs in multicellular organisms. The MHC proteins involved are the MHC class I proteins, which are molecules found on the surface of almost all nucleated cell invertebrates. Their main function is to present fragments of proteins or peptides derived from within the cell to cytotoxic T cells or to CD8 T cells. Let's move on to T lymphocytes. T cells are a type of lymphocyte which is a specialized white blood cell crucial for adaptive immunity. In simpler terms, they are like the soldiers of our immune system. They are specialized white blood cells that play a crucial role in fighting infections and other diseases. So they originate in the bone marrow but mature in the thymus gland, where they are named after. Once they trained, they circulate throughout the body, seeking out and destroying infected cells, cancer cells, and other threats. T cells play a central role in self-mediated immunity by directly or indirectly eliminating infected cells and abnormal cells. So there are different types of T cells, each with its own job, like helper T cells that coordinate the immune response, cytotoxic T cells that directly kill infected cells, and regulatory T cells that help control the immune system to prevent it from going overboard.
So we have here the types of T cells. T cells are crucial in the cell-mediated immune response because they directly target and eliminate infected or cancerous cells. They identify and destroy these cells through specific antigen recognition, playing a key role in maintaining the body's defense against intracellular pathogens. So we have here three types of T cells. First is the helper T cells, or the CD4, wherein it regulates the immune response by activating other immune cells and directing the type of immune response. And next is the cytotoxic T cells, which is the CD8, wherein it also directly responsible for cell-mediated immunity, kills infected cells or abnormal cells, like for example the cancer cells and transplant cells. Next is the suppressor T cells. It suppresses the immune response to prevent autoimmune disease and maintain immune homeostasis. And now let's dig deeper more about helper T cells. So helper T cells have a receptor on their surface called a CD4 receptor. The CD4 receptor interacts with major histocompatibility complex, which is the MHC class II molecules. MHC class II molecules sense when there is an infection or foreign substance in your body. And the CD4 receptor and MHC class II molecules activate the helper T cells. The helper T cells release molecules called cytokines. Cytokines send messages to other immune cells to start an immune response. So when helper T cells detect an infection, they form into one of two subtypes. First is the Th1 helper cells, which it releases a molecule that activates a type of cell called a macrophage. Macrophages are specialized cells that help eliminate foreign substances from your body. Th cells also activate cytotoxic T cells. And next is the Th2 helper cells, also release molecules that activate B cells. B cells create antibodies. They also release cells that cause coughing, sneezing, or diarrhea to help your body get rid of foreign substances. This group of T cells helps generate allergy antibodies. So the next type of T cells are the cytotoxic T cells. Cytotoxic T cells, also known as CD8 plus T cells or killer T cells, are a type of white blood cell that plays a critical role in the immune system by directly attacking and destroying infected or abnormal cells. It includes cancer cells and cells infected with viruses. They are identified by the presence of the CD8 molecule on their surface, which differentiates them from other T cells like helper T cells. These cells are activated by recognizing specific antigen presented by MHC class I molecules on the surface of target cells. This recognition is facilitated by their T cell receptors. For us to be able to understand more about cytotoxic T cells, here are some important events in the killing mechanism of cytotoxic T cell. First is the formation of the immunological synapse. This is a critical initial step where the cytotoxic T cell forms a tight junction with the target cell known as the immunological synapse. This involves interaction between adhesion molecules such as LFA1 on the CTL and ICAM1 on the target cell. This synapse ensures that the delivery of cytotoxic molecules is precise and directed only at the target cell, preventing collateral damage to surrounding healthy cells. Next is the release of cytotoxic granules. After the immunological synapse is established, the CTL releases cytotoxic granules into the synapse. These granules contain perforin and granzymes. Perforin forms pores in the target cell membrane, allowing granzymes to enter the cell. Once inside, granzymes activate corpuscles and other apoptotic pathways, leading to programmed cell death or apoptosis of the target cell. This event is crucial for the elimination of infected or malignant cells whilst preserving the integrity of surrounding tissues. So we have here pictures and diagram that shows how cytotoxic T cells work. So first, a virus invades a host cell. The virus produces viral proteins that become fragmented. The viral antigen is presented on the surface of the host cell alongside the cell's self-antigen. The cytotoxic T cell will bind to the antigen-presenting cell. At the time of the cytotoxic T cell binding to the antigen, the cytotoxic T cell receives a signal from the helper T cell. The cytotoxic T cell releases perforin that create a hole in the virally infected cell. The cytotoxic T cell also releases a chemical called fragmentins. Fragmentins enter the cell through the pore and cause apoptosis. The last type of T cells are the suppressor T cells. Suppressor T cells, also known as regulatory T cells, play a crucial role in maintaining immune system balance and preventing autoimmune diseases. Suppressor T cells identify potentially harmful immune responses. When the immune system detects a threat, like a virus or bacteria, it activates other immune cells to fight the invader. Sometimes this response can go overboard, attacking the body's own tissues. Suppressor T cells step in to suppress or dampen the excessive immune response. We have here some mechanisms of suppressor T cells. So first is a direct inhibition. Tregs or suppressor T cell directly interact with other immune cells to reduce their activity. Next is the cytokine release. Suppressor T cells produce anti-inflammatory cytokines. And next is its resource competition. They can compete with other immune cells for essential growth factors, limiting the resources available for those cells to proliferate and become active. Last is the modulation of dendritic cells. Tregs can influence dendritic cells, which present antigens to T cells to be less effective at stimulating an immune response.
Types of T-cell-mediated immune reactions. Its main categories are type 4, hypersensitivity, T-cell-mediated cytotoxicity, and autoimmune responses. Type 4 hypersensitivity, also known as delayed type hypersensitivity or DTH, mediated by T cells rather than antibodies. The reaction typically takes 48 to 72 hours to develop after exposure to the antigen, as shown in the illustration. This type of hypersensitivity involves a cell-mediated immune response where T cells recognize antigens presented by antigen-presenting cells, ABCs, and subsequently release cytokines that recruit and activate other immune cells such as macrophages. Mechanisms of type 4 hypersensitivity. Antigen presentation. Antigens are processed by antigen-presenting cells, ABCs, including dendritic cells and macrophages. These ABCs present the antigen on major histocompatibility complex or MHC class II molecules to CD41 T helper cells, Th1 cells. Activation of T cells. Th1 cells recognize the antigen MHC complex, leading to the activation and proliferation. Activated Th1 cells secrete variety of cytokines, with interferon gamma being particularly important. Cytokine release. LEN1 and other cytokines such as tumor necrosis factor alpha attract and activate additional immune cells, including macrophages and other types of T cells. Activated macrophages release pro-inflammatory cytokines, chemokines, and enzymes that contribute to the inflammatory response. Inflammatory response. The recruitment and activation of immune cells result in a localized inflammation and tissue damage. This immune response aims to contain and eliminate the antigen but can lead to significant tissue pathology if uncontrolled or chronic. T-cell-mediated cytotoxicity. T-cell-mediated refers to the immune response where cytotoxic T lymphocytes, CTCs, primarily CD8 T cells, directly kill infected, cancerous, or otherwise abnormal cells. Unlike antibody-mediated immunity, which targets extracellular pathogens, T-cell-mediated cytotoxicity is essential for recognizing and eliminating cells that display abnormal antigens on their surface, such as viral peptides or tumor antigens. CTLs are activated in response to antigen presentation in major histocompatibility complex MHC S1 molecules found on all clade cells. This recognition ensures that CTLs can inspect and respond to the intracellular environment of virtually all bad cells.
So for the mechanism of T-cell-mediated cytotoxicity, we have different responses or immune reactions. First one, we have this antigen recognition. So infected or abnormal cells present peptide antigens on their surface via MHC or major histocompatibility complex class 1 molecules. Then CD8 plus T cells recognize these antigen MHC1 complexes through their T cell receptor or TCR. And after this recognition of antigen, we have activation and proliferation. Upon recognizing the antigen MHC1 complex, CD8 plus T cells receive additional signals from co-stimulatory molecules and cytokines. Example, IL-2 provided by helper T cells or the CD4+. These signals promotes the activation and proliferation and also the differentiation of CD8 plus T cells into effector CTLS or cytotoxic T lymphocytes. Third one, we have the target cell killing. So perforin granzyme pathway. CTLS release cytotoxic granules containing perforin and granzymes. Perforin forms pores in the target cell membrane, allowing granzymes to enter and trigger apoptosis by activating caspases and other apoptotic pathways. And then we have this fast ligand or FASL pathway. CTLS express fast ligand or FASL on their surface, which binds to fast receptors on the target cell, inducing apoptosis through a signaling cascade involving caspase activation. Then we have this last one, apoptosis induction. The activation of apoptotic pathways ensures the controlled destruction of the target cell, minimizing inflammation and collateral damage. Then apoptotic cells are phagocytosed by macrophages, which helps clear the cellular debris and prevent the release of potentially harmful intracellular contents. So for the examples, we have these viral infections. During viral infections, infected cells present viral peptide via MHC class 1 molecules. Then CTLS recognize this peptide and kill the infected cells, thereby limiting viral replication and spread. Then we have the second one, is tumor immunity. Cancer cells often express abnormal antigens or tumor-associated antigens due to genetic mutations. So CTLS recognize these antigens and can kill the tumor cells, playing a role in immunosurveillance in the elimination of emerging cancers. We have this transplant rejection. In organ transplantation, recipient CTLS can recognize donor cells as foreign due to differences in MHC molecules. These recognitions can lead to the direct killing of donor cells, contributing to graft rejection. Moving on to autoimmune responses. So, these autoimmune responses occur when the immune system mistakenly identifies the body's own tissue as foreign and mounts an immune response against them. So this can involve both the innate and adaptive immune systems, but the adaptive response, particularly involving T cells and B cells, plays a crucial role. Then now autoreactive T cells and B cells can become activated and target soft tissues, leading to chronic inflammation and tissue damage. So for the mechanism of autoimmune responses, we have this breakdown of cell tolerance. So genetic predisposition or the environmental factors, example, infections, toxins, and hormonal influence can contribute to the breakdown of immune tolerance. So the failure of central tolerance during lymphocyte development can result in the survival of subreactive T and B cells. Failure of peripheral tolerance mechanisms such as regulatory T cells or Treg function can allow these subreactive cells to become activated. Then next one, we have this activation of autoreactive T cells. So autoreactive T cells recognize self-antigens presented by MHC molecules on antigen-presenting cells or ATCs. This recognition can lead to the activation and proliferation of autoreactive T cells, which then mediate immune attacks on self-tissues. So we have also this role of B cells and autoantibodies. The autoreactive B cells can produce autoantibodies that bind to self-antigens, forming immune complexes that deposit in tissues and trigger inflammation. B cells can also present antigens to autoreactive T cells, further than amplifying the autoimmune response. Lastly, cytokine release and inflammation. Activated T cells and B cells release cytokines and chemokines that recruit and activate other immune cells, such as macrophages and neutrophils. The resulting inflammatory response can cause tissue damage like fibrosis and loss of functions in affected organs. So for the examples, we have this type 1 diabetes, an organ-specific autoimmune disease where autoreactive CD8 plus T cells attack and destroy insulin-producing beta cells in the pancreas. Then it leads to the destruction of insulin deficiency, or it leads to insulin deficiency and hyperglycemia. We have also this multiple sclerosis, an autoimmune disease where autoreactive T cells attack the myelin sheath or surrounding nerve fibers in the central nervous system. This demyelination leads to disrupted nerve signaling, resulting in symptoms such as muscle weakness, coordination problems, and cognitive impairment. Lastly, this rheumatoid arthritis, a chronic inflammation autoimmune disease where autoreactive T cells and autoantibodies target the synovium, or it is the lining of the joints. So this results in synovial inflammation, joint pain, swelling, and eventual destruction of cartilage and bone.
Development and functions of effector T lymphocytes and cytotoxic T lymphocytes. T cell development. T cells originate from hematopoietic stem cells in the bone marrow. Immature T cells migrate to the thymus where they undergo maturation and differentiation. Key processes include positive selection, ensuring T cells can recognize MHC molecules, and negative selection, eliminating self-reactive T cells. Effector T lymphocytes. These are activated T cells that perform immune functions. Two types include helper T cells or CD4+, assist other immune cells by secreting cytokines. Next, cytotoxic T cells or CD8+, directly kill infected or cancerous cells. For its activation, naive T cells are activated by antigen-presenting cells or APCs presenting specific antigens. Activation and differentiation. APCs present antigens via MHC molecules to T cells in lymphoid organs. There are three signal requirements. Signal 1, antigen recognition by T cell receptor or TCR. Signal 2, co-stimulatory signals provided by APCs. Signal 3, cytokines that guide T cell differentiation. Activated T cells differentiate into effector T cells or memory T cells. Helper T cells or CD4+, secrete cytokines to regulate and assist other immune cells. There are four subtypes. Th1 cells promote macrophage activation and are important for intracellular infections. Th2 cells assist B cells in antibody production and are involved in allergic responses. Th17 cells enhance neutrophil response and are involved in inflammation and autoimmunity. Treg cells suppress immune responses and maintain tolerance. Cytotoxic T lymphocytes or CDLs, they are also called CD8+, are effector T cells specialized in killing infected or cancerous cells. For its activation, they recognize antigens presented by MHC class I molecules on infected or abnormal cells. So the two mechanisms of killing are the following. First, we have perforin granzyme pathway. Release of perforin forms pores in the target cell membrane, allowing granzymes to enter and induce apoptosis. Next is the fast-fast ligand pathway. CDLs express fast ligand that binds to fast receptors on target cells, triggering apoptosis. Functions of CDLs. CDLs recognize and kill cells infected with viruses, limiting viral replication. Tumor cells. CDLs target and kill cancerous cells expressing abnormal antigens. Graft rejection. CDLs can recognize and destroy foreign cells in transplanted tissues, contributing to graft rejection. Memory T cells. Long-lived T cells that provide rapid and robust responses upon re-exposure to the same antigen. The two types include central memory T cells, or TCM, reside in lymphoid tissues and provide a quick response upon antigen re-exposure. Effector memory T cells, or TEM, circulate in the bloodstream and rapidly respond to infections in peripheral tissues. Conclusion: the cell-mediated immune response is indispensable for effective defense against intracellular pathogens, tumor cells, and in regulating immune responses. A healthy cell-mediated immune response is crucial for fighting off various infections, controlling chronic conditions, and maintaining overall health. Continued research and advancements in immunotherapy hold promise for harnessing the full potential of T cells to treat various diseases, highlighting the vital role of cell-mediated immunity in maintaining health and combating disease. Thank you for listening.